Very good morning. So today I'm taking up one of our major topics in the muscular system, and this is the muscle organization. So you can get a full-fledged question on muscle organization in your final papers. So first of all, I should tell you some basic concepts in muscle organization, that what is a muscle belly or the muscle organ. So if we take out a full muscle out of the body of some animal with these are the tendons on both the sides. So this whole structure, the whole muscle, it is called as a muscle belly. It is also called as a muscle organ. So do not confuse in between these two words. So the word muscle belly is used for whole of the muscle along with the tendons. Okay, now we have to go inside this muscle organ to see the organization of the muscle. So the skeletal muscles, when they are taken out of the body of the animal, they are ensheathed by a tough layer of connective tissue, which is called as epimysium. Epi refers to the outermost, and mysium or mycia are the layers of the connective tissue which are covering the muscle or the muscle belly. So epi is the outermost connective tissue layer. Now this epimysium, it anchors the muscle tissue to the tendons at the each end. So we can say that the muscles are wrapped inside this epimysium, and this wrapping is continuous with the tendons on both the sides. So if I redraw it, suppose we have a muscle. So this is a full muscle and it is covered by epimysium. This is the connective tissue, and this connective tissue is continuous with the tendon on both the sides. So that means tendons are also a connective tissue structure. It is also wrapped in the connective tissue, the muscle. So this is a continuous sheath over the muscle which is enclosing the whole of the muscle organ. So epimysium is the outermost layer of the connective tissue. Now it also protects the muscle from the friction against other muscles and bones. So this outer layer, it also helps to protect the muscle from the friction. So within this epimysium are the multiple bundles of the muscles which are called as fascicles. Now if we take this muscle inside the epimysium, this is containing a large number of muscle bundles which are called as muscle fascicles. One is known as fasciculus. Now each of these fasciculus may contain like 100 muscle fibers. Now if we go inside a fascicle, it will contain here about 100, it may be like 10 to 100 muscle fibers. So fascicle is the biggest bundle that we can have in a muscle, and then we have the muscle fibers, and within muscle fibers we can have the myofibrils. Now one thing to note is that the individual muscle fibers, they are ensheathed by endomysium. Whereas all the fascicles, they are lined by perimysium. So whole of the muscle is lined by epimysium, then the fascicles, they are lined by perimysium, and if we take the muscle fibers, they are lined with endomysium. So in these myofibrils, if we go beyond inside the myofibrils, we have sarcomeres, and sarcomeres, these are the smallest unit of the muscle which can contract. Now if we see the structure of the sarcomere, it is containing dark and light bands. So these are the dark bands, these are the dark, this is a dark band, this is a dark band, and this is the light band. So these myofibrils are striated in nature, and you can very easily see these dark bands. And then you have Z lines in the light bands, and in between these two Z lines, this part is one sarcomere, this is the second sarcomere, this is the third sarcomere, and so on. So one myofibril can have hundreds of sarcomere units. So this is the one unit which can contract on its own. So if we go beyond this in sarcomeres, we can say that we have two types of proteins, major two types of proteins. One is myosin, other is actin. One is alpha actinin, one is nabulin, and one is titin. So these are the 5 kinds of proteins which are present in the sarcomere. So major you are dealing with the actin and myosin. In this diagram, I am teaching you the organization of a muscle, which we have discussed earlier. So this is the whole of the muscle belly. You can see this is the outermost layer, the epimysium. This is the covering, the connective tissue covering of the whole muscle belly. And if we go inside this epimysium, we have got, these are the fascia or the fascicles. So this is a muscle fascicle, and these are the large number of fascicles inside this muscle belly. Now if we take out a single fascicle out, now we are taking this fascicle out. So one fascicle has got many muscle fibers. So these are the muscle fibers, and these muscle fibers, they have got many myofibrils. Now if you see, this is a fascicle, this is a single fascicle out of this muscle belly. It is unsheathed by a layer which is called as perimycium, and inside this perimycium we got many muscle fibers, and these muscle fibers, they have got myofibril. So these muscle fibers, they are again ensheathed by endomycium. So this single muscle myofibril, it has got dark and light bands, and if we zoom into it, we can see the sarcomeres, and sarcomeres has got thick filaments and thin filaments, which are composed of myosin and actin protein. So these are the Z bands in between the light bands. So in between these two Z bands, we are considering it as a single sarcomere. So the length in between the two Z lines is called as the sarcomere. So this is the structure of a single sarcomere. So if we go in the hierarchy of the muscle organization, first of all we have got a full muscle belly, then we have the fascicles, then we have muscle fibers, then we have myofibrils, then we have sarcomeres, and then we have myosin and actin protein.